ちょっとテント買いました。ちょっとこれ試しに広げてみましょう。これは撮ってみますかね。あるんだ。
We know a lot about the parts of the cell, but we really don't have a good model for how they're integrated together. And omics, functional genomics, is what tries to do that in a quantitative way. It's not that we're very good at it at this stage, but it's sort of the beginnings of it, and our project has kind of contributed to this. So, I'm going to talk about a bunch of us work on here. Georgina and David are leaders in the field, and it's a eukaryotic cell, so it's a billion years further evolved beyond prokaryotes, and it's got a nucleus and mitochondria with its own DNA. It's got all the goodies that we find in human cells, and so it's been a major model organism. And in fact, there's 1,000 yeast labs around the world, and we all feed data into one big common database that is put together at Stanford by SCD. So yeast has 6,000 genes and 16 pairs of chromosomes, and you compare that to a human has 20,000 genes, 23 pairs of chromosomes. So it's a pretty sweet little cell. And the yeast community, this international community, there's labs all over Europe, major labs in Japan, and of course in North America, and it's expanding to China and India as we speak, not in Russia. But anyway, the goal is to figure out, and we've got a graduate student, there's 6 graduate students, we've got a graduate student for every single yeast gene, and you'd think that we could figure out how this thing works, this cell works tomorrow, but we're so far from it, because it's really good how they're all connected to each other and how they're talking to each other and the dynamics of it all, and that is way beyond the yeast studying any form of life. But just to give you a view of that, here's 16 chromosomes, each line represents a gene, so these genes are just lined up one after another on each chromosome, and one thing about these because it's so like this organism genetically, probably through various recombinations, we can go into nucleotides, so there's 2 copies of the chromosome, and delete one copy of each gene. And then the beauty of yeast is you can take it and convert it into a haploid, and where it only has 1 copy of each chromosome, and you can under those conditions ask whether that gene is required for life. And so the yeast community did this, deleted each of the 6,000 genes one by one in a diploid, converted it into a haploid, and asked whether the gene is required for life. Basically barely knows it's happened. Okay. And this is true not just in yeast, it's true in all forms of life, basically, any sophisticated form. So in E. coli, in yeast, in organism flies, in mice, and in human cells, only a fraction of the genes are required for the basic cell division and the replication of chromosomes. Most of the genes appear to be functionally redundant, okay, so that you can delete them, and they must be doing something because they're highly conserved, but it looks like there's a backup plan for whatever it is. So a nice example is DNA repair. So control DNA repair different pathways, and they're required under particular conditions, but in a laboratory setting, you can remove non-homologous end joining, and the cell's perfectly fine. It can repair its DNA through homologous recombination, for example. But if you remove both those pathways, there's non-homologous end joining, then the cell has a problem. So we think many of the reasons these cells aren't required for viability is because the cell is wired with backup systems, and so that the analogy is a finely tuned jet engine where if something goes wrong, you got another way to keep the plane up in the air. So.
Why, maybe it's philosophical, but why would the cells of Robotnik work out remaining essential genes and have backup?
That's a good question. I can't figure that out, except to say that, it wants to streamline everything as much as possible, but still, then why wouldn't there be a backup pathway for that thing? There's really only one proteasome. It doesn't make another copy of all those genes and another. There's other ways autophagy and things to degrade proteins, but the major essential complexes are things like the ribosome, the proteasome, and the whatever.
To make sure you're not telling us that you had a yeast cell where you deleted 5,000 genes and the remaining thing is alive, right?
No, but that's not so. Craig Venter tried to make the minimal genome, right? And basically, if you read their paper, they tried to take the equivalent of these 1,000 genes, and see if it would work. And then they were really surprised when it didn't work. So yeah. Do you want to give me any feedback? He did bacteria, but they basically the idea was this: if you read the paper, they're really surprised to rediscover synthetically validate. I don't think it's the best way to think about it. And it's way more complicated than just one pathway backing another one out. When I show you, so it's just a very simple beginning thought that's not correct.
I would say something that, for example, some of the genes become essential once given other conditions of substrate. So now they become essential. So they're all essential. So when they're essential, these are more essential genes. So the genes are really higher. You can see why they want to go there. The outcome they have to split them.
It's complicated, right? Yeah. Okay, so anyway, so here's what we're going to do. We're going to explore gene function through this genetic interactions, and this is a term that does not mean interaction physically. Okay, it just means some kind of more like a mental interaction. Sex. No, I don't know what it means. And but here's a definition. A genetic interaction occurs when mutations in we'll just focus on two right now, but it could be more. Two different genes combine to generate an unexpected double mutant phenotype. Okay. So if I have mutant A and I know what it does to the cell, and I have mutant B and I know what it does to the cell, when I put those two together, I expect to see both things happening, right? That's the additive effect of the generated. But we call it genetic interaction when I take this and put together, then something crazy happens, right? Something unexpected. So the guy that we all know and love that articulated this in the Yeast community was this guy, David Botstein, and he really did articulate this when you go back and look at the literature, and which is kind of surprising because we think that the Yeast community thinks it's been looking at genetic interactions for years and years and years, but it wasn't actually. I'm almost going to graduate from my PhD at this point, and this paper from famous Novik and Botstein comes out, and they're looking for suppressors. This is a genetic interaction of yeast actin mutations, and so. This is a yeast cell stained with fermentous actin with rotase. That's a good picture. Yeah. I know. So, of course, David is the yeast expert. But look at that. The mother cell, the daughter cell. We've got these patches here. David's going to talk about. They're all involved in endocytosis. And then these cables. And this only has one actin gene. Okay, so this makes it, and it's essential. If you delete it, you don't have any of these guys, the cell can't form any shape properly, and it can't divide. But and if you go to a worm, and nematode, they have lots of different actin genes, and so you can't really do this type of experiment anymore. So what Novik and Botstein did was they mutagenized the actin gene, and this -1 means it's a particular allele, so some particular mutation, and they isolated a mutant that was alive at 23 degrees and dies at 37. Okay, so this is a temperature-sensitive mutant, and yeast and fly geneticists use this to study essential genes in a conditional way, so we can grow them at a permissive temperature and then shift to the restrictive temperature and see what happens, reveal the essential phenotype. And one way to think about it is that there's lots of different things that happen. The protein might not be made at the restrictive temperature, it might be degraded, but the classic idea is that you have a mutation in the polypeptide chain that allows it to fold normally at the permissive temperature, but when you put energy into it, the protein misfolds and you reveal the mutant phenotype. There are examples of that. So what they did was they looked for suppressors of actin mutants, and so what happens here is if you combine ACT1-1 and you basically select for a random mutation in the genome that suppresses this temperature sensitivity, and you identify a new gene that way, and so ACT1-1 plus this mutation, the cells are alive at the restrictive temperature, and we'll call this this is an example of genetic expression. We'll call it genetic interaction because it is an unusual phenotype. You go from dead to alive, right? And that's pretty powerful. And since it's a gain of a phenotype, we call it positive genetic interaction. So when they did this, it was completely unknown. They wouldn't even have known what this gene is. It turns out that it's involved in phosphatidylmyosin signaling, which is a signaling pathway that can regulate actin dynamics through other proteins. We can probably still know the mechanism, but there's some major function between these two genes. Okay. And then this is not that case, but we've been one has 6. The cells are dead, even at the permissive temperature. Okay. And again, I'm just kind of trying to look at the mutations. I don't think we understand any of this, right? We should look it up though and try and figure it out. Anyway, so this is an example of so this is a rescue positive genetic interaction. This is synthetic lethality, which is a negative genetic interaction. So, and this is a very famous one that we're going to talk a lot about. But this is unusual. Like mutation, this is viable at 23 degrees. This one's perfectly viable at 23 degrees, but the double mutant is dead. Okay. And this would say to us that these two genes are working together to control the central function in the cell, right? And presuming that's actin assembly, and this mutation compromises the actin gene itself, and then this mutation must, exacerbate that and somehow have something to do with all the whole actin dynamics. Okay, so synthetic lethality is a very simple definition where it deletes one gene and the cells are viable, delete another and they're viable, but the double mutant is lethal, and this is a rare event. If you take DNA and test all possible double mutants, you'll only find a few cases like this. And because it's rare, geneticists thought that this must be important and it must be telling us that these two genes are working together somehow in the cell. And what David did was he went into the literature and figured out that this combination of two mutations leading to a dead phenotype has a name called synthetic lethality, and it was coined by this guy Jansky, who was a phylogeneticist in the 40s. And so synthetic lethality occurs when the combination of two mutations by itself causes lethality. And the wild thing is that Jansky was working with flies from an outback population, so he was going out into the wilds of California, isolating flies, crossing them to one another, and he could trace a defect in development to an allele of one gene from one parent and a allele of another gene from another parent. And because he's doing complete genetics, this could not have been trivial, but he was seeing it. And there's a couple implications there. One is that it occurs not just on extreme mutant alleles that we make in the lab, it occurs on wheels of genes that are out there in natural populations that carry natural variation, and therefore it may underlie a lot of genetics as we know it. So genetic interactions might underlie a lot of genetics. So anyway, there's a lot of combinations of genes. So there's 6,000 yeast genes, so there's 18 million gene pairs. And maybe I'll talk about triplets, but there's 36 billion gene triplets, right? So when we start thinking about genetic interactions, combinations of alleles driving inherited phenotypes, there's huge potential for this to take place in any genome. So John Pringle was in his famous yeast test at Stanford now. He was in Hubbard's lab where they developed the Segway assay, and along with Alan Bender, they carried out the first synthetic lethal screen. And so this works. You can set up a plasmid with a gene on it, and you delete that same gene from the genome, and then you mutagenize the cells, and you look for secondary mutations that make that plasmid essential for viability, so the cell can't lose the plasmid. And so, this guy can lose the plasmid, this one cannot, and so it's probably carrying a synthetic lethal mutation with the query strain that they were looking for. So they started off with query mutations in BEM1 and BEM2 that are involved in divergence, and they mutagenized those strains with this plasmid loss thing, and they would find synthetic lethal interactions with MSP1. And this was the first screen. It took probably a couple years to find that one synthetic lethal interaction because of that work. And so the beauty of this set of viable deletion mutants is that it opened the door for systematic genetics. And so we could now have a matrix of 5,000 mutants, and we could cross any mutation into that whole set and make 5,000 double mutants. Because we can make temperature-sensitive alleles of the essential genes, we can also look for synthetic lethal interactions, just like David Botstein and Pierre Novick did by mutating essential genes, getting TS alleles, and then doing crosses at a permissive temperature where the cells are still alive, but they carry a mutation that compromises the essential gene function. And so this is where Brendan and I got together because we started to think, we knew that the yeast community was creating this reagent set, and we started to think, well, the double mutants are going to be the most fun, and so we're trying to set up a system to make double mutants, and we can do it because yeast genetics is just so easy to do, and so I often wonder, why did I spend 5 years of my life studying yeast mating as a young adult, when I should have been studying money, right, and making money, like my brothers did, but in the end, you study mating when you can figure out how to make lots of double mutants, so I can make millions of dollars by making millions of double mutants, and so then we can explore gene function with synthetic lethality, so you can cross this and find these things. And so the way we think about it, and again, this is just a model, is that if you have two pathways like homologous end joining and homologous recombination and non-homologous end joining, and they both impinge on the central function, you can tolerate a mutation in one because the other's still functioning, or this one, the other's functioning, but a double hit will kill the cell. Okay, so this is a synthetic lethal interaction, and it often occurs between sets of genes that are encoding molecular machines that work together to drive essential functions. So if I remove one component of this machine and I screen all other genes for synthetic lethal interactions, I'll identify identify synthetic lethal interactions with all the components of the backward pathway. And that's true for this component and this one. So that genes that have similar patterns of genetic interactions are in the same pathway or complex. And so given protein complex, we can often see if these two complexes are working together, we'll see coherent sets of synthetic lethal interactions between all the components of the complex. And they can be strong or they can be weak, but they're all coherent, so we know they're true, they're statistically real. So Lee Harlow won the Nobel Prize for his cell division cycle. He was really interested in synthetic lethality, and he articulated these two concepts around 2000, so about 20 years ago. One is that if you have a cancer pathway that's it's a non-essential gene set, and you can then kill cancer cells specifically by targeting synthetic lethal interactions that take advantage of those vulnerabilities in the cancer cell. Okay, and that works like mad. Everybody's going nuts with this now, because when he talked about this, you couldn't do any kind of genetics in human cells, but with CRISPR, it's just so painfully obvious that this can be done. Harlow was arguing you should do it in yeast and worms and flies. If it holds true, then we'll go and test it in mice and maybe human cells somehow. But now you can do screens like this and find it. But then the other idea he talked about, which is even wilder, is this genotype to phenotype potential. And so he was thinking of, okay, now we're going to start sequencing everybody. We're going to get their whole genomes, and then we have to interpret it, and we'll have to interpret natural variation. And if natural variation can combine either digenic interactions or trigenic interactions to modulate pathways and lead to inherited phenotypes, we have to know the rules of how genetic networks work, right? And we have to figure out how prevalent it is. So, and that's a wild idea. I dropped the Kool-Aid because we made our grants and we cite Lee Harlow's paper all the time, and we say this is why you should give us money to do this. But there really is almost no evidence of genetic interactions associated with human genetics. And the reason is because it's a statistical game that we can't win. 20,000 genes, 200 million possible gene pairs, and if you look for, say 1,000 people in a GWAS study, you just don't have enough statistical power to find a single genetic interaction. And even more than that, when you think that if the genetic interactions are mapping, say, two different pathways that impinge on something that's important for disease state, then there's many different pairs of genes that will lead to the same phenotype. So you're trying to find multiple different pairs of genes that are associated with a disease state, right? And so that's another thing. And I explained there. So there's this missing heritability for any human disease, and this is where I don't know what I'm talking about very much, except for what I'm saying. So bear with me on that. But for any disease you look at, usually we only can explain about 20 to 30% of the heritability. A lot of it's missing, and there's many different thoughts about this, that it could be rare mutations, blah, blah, blah. We just don't know what's going on. But it's quite possible, and it looks from yeast models, that definitely genetic interactions will be some significant component of that. That being said, there's almost zero evidence for it. And he'll kind of walk through this. This is why instead of calling it missing heritability, he's calling it heritability, because if the variation is there, we've documented it, we just don't know which combinations are leading to the phenotype. So he sort of goes through the math and says to find one particular genetic interaction, you'd have to have around 500,000 individuals. So someday we'll have that when our sequences are on our phone and all our records are being monitored by Google or something. But there's other ways maybe to get at this, and our collaborator Chad Myers has some ideas, and if I get to that, we can talk about it. But so anyway, we started based on Harlow's suggestion, basically, and just the pure thought of it, we decided to try and make all possible different solutions. Thank you. I'm definitely going to skip something, but but I can go to the highlights. Anyway, we really, Amy Tong was a graduate student who started this. She's on this picture, but then Michael Costanzo took over. We have a team of technicians, and they feed these locally designed machines to just make all the double so we can just replica plate, and it works. The computational analysis is the tricky part, and Chad Myers was in the Boulder Troy Skyers lab at Princeton, and he has a team of postdocs, graduate students. Anastasia was a graduate student with Ben and I, and she's now at Calico, and these guys really formed a team. And it's very simple. Our model for how to score a genetic interaction, if you delete a gene and you reduce the growth rate, but you don't kill the cell by a defined amount. So that's this one, reduce it to 0.7, delete this gene, reduce it to 0.5. If I delete this gene in this background, I expect to see a reduction of fitness by 0.5. Okay, that's our little model. So the fitness is like the combination of the two single mutants we expect to lead to a phenotype of 0.35. So synthetic value of the double mutant is dead. And that's making genetic interaction. But if the double mutant goes better than we'd expect, we call that positive genetic interaction. So we want to find negative and positive genetic interactions. And we do it just by looking at colony size on a plate. And there's all kinds of systematic errors that we go from noisy data to data where we normally genetic interactions. So then basically what we get is a vector of negative and positive genetic interactions. So I delete gene A, and if it shows a positive genetic interaction with B or with C, or a negative for these other guys, we score it and we quantify it. And we do that we cross gene A to all 6,000 other mutants, and then just score these things. And you get basically, here's an example. So RAD52 is involved in DNA repair, and we have negative interactions with all these other genes that have very well-defined roles of DNA synthesis and repair. So you connect, in general, functionally related genes. I mean, there's noise in the data, and if the gene is a signaling molecule that controls